Hello everybody, I am Dr. D. Kavita, Assistant Professor, Department of Biochemistry and Biotechnology, Nashingam Institute for Home Science and Higher Education for Women, Coimbatore. Now you have chosen to study the module on fermentation technology in Food Biotechnology paper. This module describes the fundamentals of fermentation technology, a multidisciplinary area of biotechnology which involves the skills in science and engineering. Fermentation technology is ubiquitous. They range from the production of food in a kitchen to the synthesis of sophisticated, extremely high-value, cutting-edge therapeutics. The principle of bioprocess technology involves the use of cells as microfactories to manufacture the product of interest. The cellular products can be directly utilized, or alternatively, the cells can be engineered to produce products of interest. However, the production levels can be improved by employing several strategies for formation or processing of the product. Coming to the objectives of this module, you will understand the steps in the bioprocess for the production of products using integrated biochemical processes. You will be able to convert the promises of molecular biology and genetic engineering into new processes to make commercial products in economical, feasible way. Let us start by defining the fermentation technology. Fermentation technology is defined as any process that produces a product by the mass culture of a microorganism. Also, its biochemical meaning is an energy generation process in which organic compounds act as both electron donors and terminal electron acceptors. The cultivation of microorganisms to produce useful products or to undertake useful activities is, however, not a recent invention. Hence, we shall look into the history of fermentation technology. Fermented foods such as yogurt, cheese, soya sauce, pickled cabbage have been intentionally produced by man for centuries. Beer recovered from the pyramids of the ancient Egyptians reveal that the Egyptians were able to produce the fermented brew using almost pure cultures of yeast. In Renaissance Europe, the nitrates required for gunpowder manufacture were produced by allowing soil bacteria to nitrify ammonia in horse urine as it was flowed down channels dug into hills. The Aztecs of Mexico cultured spirulina, a single-celled algae, had been used for both waste treatment purposes and as a source of protein. In the 20th century, biotechnologists have tried again to produce single-celled protein to solve the world's protein shortage. The father of modern fermentation technology was, however, Louis Pasteur. This 19th century scientist discovered or perhaps rediscovered that the conversion of sugar to alcohol, as in beer and wine production, and the conversion of sugar to lactic acid, as occurs in cheese production, were both microbiological conversions and both needed a microbial seed to start them. In the process, Pasteur disproved the theory of spontaneous generation, discovered that some microbes can grow in the absence of oxygen, and discovered a new way of sterilizing materials which was subsequently called pasteurization. Following Pasteur, microbiology and fermentation technology grew hand in hand. Alexander Fleming's discovery of penicillin eventually led to the development of a huge antibiotic industry. The world wars and their aftermath of the 20th century continued to spur on developments in fermentation technology. Material shortages forced countries to look to biological sources of chemical feedstocks. Scientists found ways of producing or enhancing the production of ethanol and solvents such as acetone and butanol by fermentation. Antibiotic production was also scaled up to meet growing needs derived from diseases and injuries. The designs and technologies developed for improving antibiotic production found application in many other products such as amino acids, example lysine and glutamic acid, and food chemicals, example gluconic acid and citric acid. The engineer and the scientist now had to work hand in hand to achieve these improvements. In more recent times, threats of diseases, environmental disasters, food shortages are juxtaposed by growing demand in developed countries and have created new opportunities for fermentation technologists. Today, the fermentation industry is a multi-million-dollar industry. Now, let us discuss the principles of fermentation. In developing a product, a fermentation technologist must consider all stages of the manufacturing process. At the heart of the process is the fermenter. Large-scale cell cultivation is performed in specialized reaction vessels known as bioreactors or fermenters. In the fermenter, cells convert raw materials or substrates to products and to more cells. The conversion of substrate to more cells is a major advantage of fermentation processes over enzymatic and chemical processes. The production of more cells means that during a fermentation, the concentration of catalyst, in this case the cells, can increase with time and the process can actually speed up with time. The major types of cells as cellular catalysts used in fermentation processes includes the following: animal cells, namely Chinese hamster ovary cells, hybridoma cells, insect cells, etc. Moles, Penicillium species, Aspergillus species, etc. Yeast, Saccharomyces and Candida species, bacteria, Escherichia coli, Bacillus species, Pseudomonas species, Streptomyces species, and so on. Now, let us discuss the types of fermentation processes. The commercially important fermentations are divided into 5 major groups. 1. The process that produce microbial cells. 2. The process that produce microbial enzymes. 3. The process that produce microbial metabolites. The process that produce recombinant products. And the last, the transformation process. Apart from these types of processes, any established fermentation technique may be divided into 6 basic component parts. Media formulation for culturing the process microorganism, the sterilization of the medium, fermenter and accessories, active and pure culture production for inoculation in production vessel, growth of the organism under optimum conditions for product formation, extraction and purification of the product, effluent disposal produced by the process. The interrelationship between these 6 phases are shown in the picture. Now, let us discuss the various steps involved in studying a fermentation process. The process will initially be studied in laboratories using flasks and laboratory scale fermenters. If the process looks commercially successful, the process will be scaled up to a pilot scale process. During this stage, the process will be looked at in terms of engineering factors such as mass transfer and heat transfer. Downstream processing will also be looked into. The pilot scale studies will determine the commercial viability of the process, and if successful, the process will be scaled up to industrial or commercial scale. Some fermentation processes, however, can involve the complex interactions of biological, chemical and physical factors. To properly investigate a fermentation and to be able to predict the effects that these factors play on fermentations, the process needs to be broken down into meaningful units. We shall look into the various modes of fermenter operation. The fermenters can be carried out as batch, continuous and fed batch processes. First, let us see the batch culture. Batch culture fermentation is a closed system where the required amount of nutrients and other additives are added at the beginning of the process. Once the fermentation process has started, there is no refill of nutrients and the product is recovered only at the end of the process. At the end of the fermentation, the contents are removed for downstream processing. The reactor is then cleaned, sterilized and refilled for the next fermentation. During the process, the inoculated microorganisms will pass through a number of phases, namely lag phase, log phase, stationary phase and decline phase. First, there is a period during which growth does not occur, referred to as the lag phase. Lag phase is a time of adaptation. Following the lag phase, the growth rate of the cells gradually increases. The cells grow at a constant and maximum rate and the period is known as log phase or exponential phase. Eventually, the growth rate has declined to zero and the cells enter the so-called stationary phase. After a further period of time, the culture enters the death phase and the viable cell number declines. During the exponential phase, certain products are produced that are essential for the cell growth, namely amino acids, nucleotides, proteins, lipids, carbohydrates, etc. These primary metabolites are produced during the log phase. Hence, it is the task of the industrial microbiologist to improve the productivity of these primary metabolites. The secondary metabolites are synthesized during the stationary phase. Thus, batch fermentation can be used to produce biomass, primary metabolites and secondary metabolites. The second type is the continuous culture. In continuous cultures, fresh media is continuously added into the bioreactor and at the same time, bioreactor fluid is continuously removed. The cells thus continuously propagate on the fresh medium entering the reactor and at the same time, the products, metabolic waste products and cells are removed in the effluent. Continuous cultures have, however, been with us almost since life began on good old Mother Earth. Our guts are in many ways a continuous culture. Food comes in, gets processed by microorganisms and enzymes. Some is absorbed, the rest is excreted. The important feature of this technique is that the microbial growth in a continuous culture takes place under steady state conditions. That is, growth occurs at a constant rate and in a constant environment. Many factors such as pH, concentrations of nutrients, metabolic products and oxygen, which inevitably change during the growth cycle of a batch culture, are all maintained constant in a continuous culture. Today, continuous cultures find wide application in the waste treatment industry, although to date limited application in fermentation industry. Continuous culture systems have many advantages over batch reactors, but unfortunately a few critical disadvantages also. While comparing the batch and continuous cultures have several advantages over the batch cultures. It is not necessary to shut the continuous fermenter down as frequently as a batch fermenter. At the end of a batch fermentation, the reactor must be emptied, cleaned, sterilized and refilled. The time required for these operations is known as turnaround time. Theoretically, a continuous fermenter could operate indefinitely without having to be shut down. In practice, however, this is not possible because continuous cultures are shut down with less frequency as compared to batch reactors. There is less loss of productivity during lag phases. Most downstream processing operations are most productive when operated in a continuous manner. Using a continuous culture allows the fermentation to be in time with other operations in the plant. Thus, overall, plant productivity is easier to optimize. The disadvantage of a continuous culture is contamination of a continuous fermenter can have disastrous consequences. A contaminant can cause the washout of the resident organism and then completely take over a fermenter. Next, the third type of technique is fed batch culture. In a fed batch fermenter, the feed is continuously added until the maximum liquid fermenter volume is reached. The fermenter may then be allowed to continue or be partially or completely emptied depending on the process. Although low nutrient and substrate concentrations can be maintained in continuous cultures, as we have seen, continuous reactors do suffer from a number of significant problems. In fed batch reactors, feed is added but effluent and cells are not removed. Thus, fed batch reactors can be used to maintain cells under low substrate or nutrient conditions without washout occurring. Fed batch reactors are thus primarily used for producing products under low nutrient or substrate concentrations and play an important role in industry. Now, let us see the comparison of fed batch and continuous bioreactor. With both fed batch and continuous fermenters, it is possible to set and maintain the specific growth rate and substrate concentration at optimal levels. The major physical difference between a fed batch and continuous fermenter is that the effluent is not continuously removed and thus washout does not occur. This provides a fed batch reactor a number of advantages over continuous reactors. Because cells are not removed during the fermentation, fed batch fermenters are well suited for the production of compounds produced during very slow or zero growth. Unlike a continuous fermenter, the feed does not need to contain all the nutrients needed to sustain growth. The feed may contain only a nitrogen source or a metabolic precursor. Contamination will also not have the same dramatic effect on a fed batch fermenter, as a contaminant will not be able to completely take over the fermenter unless the contamination occurred during the early stages of the fermentation. Of the three systems, fed batch bioreactors are most commonly used to produce biological products. Batch reactors are the second most commonly used. Although continuous reactors are rarely used for large-scale production of biochemicals, they are widely used in waste treatment processes. In addition, natural ecosystems such as the rumen and gut, soil and rivers are more closely represented by a continuous culture system. With all this information, let us discuss the process of screening of industrially important microorganisms. The tasks of both discovering the new microbial compounds and improving the synthesis of known strains have become more and more tedious. The microorganisms of industrial importance are generally bacteria, actinomycetes, fungi and algae. These organisms occur virtually everywhere. Example, air, water and soil on the surfaces of plants and animals and in plant and animal tissues. But most common sources of industrial microorganisms are soils, lakes and river mud. There are three important stages in the screening strategies, namely isolation, preservation and improvement of the strains of commercial importance. We shall discuss the first step of isolation in screening strategies. Isolation, it involves obtaining either pure or mixed cultures based on their production of industrially relevant products. A variety of complex isolation procedures have been developed, but no single method can reveal all the microorganisms present in a sample. Specialized enrichment techniques are used to isolate many different microorganisms. Selective pressure can be used in the isolation of organisms to favor the growth of the desired types or addition of inhibitors to destroy the undesired strains. A set of highly selective procedures which allows the detection and isolation of microorganisms producing the desired metabolite constitutes primary screening. Ideally, primary screening should be rapid, inexpensive, predictive, specific but effective for a broad range of compounds and applicable on a large scale. Primary screening is time consuming and labor intensive since a large number of isolates have to be screened to identify a few potential ones. Rapid and effective screening techniques have been devised for a variety of microbial products which utilize either a property of the product or that of its biosynthetic pathway for detection of desirable isolates. This table shows the several screening approaches used for detection of microbial isolates producing some useful products. Some of the screening techniques are relatively simple. Example, extracellular enzymes, enzyme inhibitors, antibacterials, antifungals, antiviral and antitumor agents. Suitably designed specialized screening techniques may be used to detect compounds having various pharmacological activities other than antibiotics. Therefore, it is necessary to preserve the isolated strain without losing its desirable characteristics. The isolated cultures can be stored by various preservation techniques. 1. The storage at reduced temperature. If the cultures are subcultured at 6-month interval, it can be stored on agar slopes in refrigerator at 4 degrees centigrade or in freezer at -20 degrees centigrade. The time of subculture may be extended to 1 year if the cultures are covered with mineral oil. For the long-term preservation of cells that do not survive freeze drying, liquid nitrogen, which is having the temperature of -156 degrees centigrade to -196 degrees centigrade, is the suitable method for preservation of valuable stock cultures. This technique involves growing a culture to the maximum stationary phase and then resuspending the cells in a cryoprotective agent such as 10% glycerol. Next, storage in a dehydrated form. The dried cultures are commonly used especially for sporulating mycelial organisms, namely the soil. Lyophilization technique is used to store the culture in refrigerator for 10 years. This is done by freezing the culture followed by its drying under vacuum leading to the sublimation of water. The industrial cultures must be stored to eliminate genetic change, protect against contamination and retain viability. The next important step is the improvement of microbes. Wild-type microbes usually produce a very low compound titers. Therefore, in order to increase the productivity of the wild strain, various improvement strategies are adapted. Traditional methods for strain improvement are random mutagenesis and selection. The selected strains are usually subjected to successive cycles of mutagenesis and selection. After After several cycles, a large increase in yield is likely to be obtained. Such a random approach is often a good start and sufficient to generate a high producing strain. Increased yields may be achieved by optimizing the culture medium and growth conditions, but this approach will be limited by the organism's maximum ability to synthesize the product. The potential productivity of the organism is controlled by its genome. Therefore, the genome must be modified to increase the potential yield. The techniques and approaches used to genetically modify strains to increase the production of the desired products are called strain improvement or strain development. Genetic modification may be achieved by selecting natural variants, by selecting induced mutants, and by selecting recombinants. There is a small probability of a genetic change occurring each time when a cell divides. Hence, a microbial culture will undergo a vast number of such divisions. The culture will become more heterogeneous. However, variants have been isolated which are superior producers, and this has led to the development of a natural product from a newly isolated organisms. Recombinant DNA technology has enabled the production of heterologous products and has built on the achievements of directed selection to increase the yields of conventional products still further. Thus, this module has presented the overview of fermentation technology, where the microorganisms are capable of growing on a wide range of substrates and can produce a spectrum of commercially valuable products. Bioprocess technology deals with various biotechnological processes used in industries for large-scale production of biological product, for optimization of yield in the end product and the quality of the end product. Further, the bioreactor design and downstream processing will be discussed in the next modules. Thank you.